Olivier Belli, welcome to the podcast.
Well, thank you very much for having me.
It's a real pleasure. You're joining us from Switzerland, right?
Yeah.
Yeah. Now, just before we get started, a little bit about you. It's my understanding that you're originally from France, right?
Yeah, I'm French, so I work in Switzerland in Basel, which is basically right at the border with France. So I'm close to home.
Okay, where did you grow up?
I work in a small town called Metz, close to Luxembourg. So yeah, northeast of France.
Oh, okay. So is that where you grew up?
Yeah.
Oh, okay. Okay. So you're very familiar with the area that you're in now. Cool. And did you always want to study science?
Yeah, I think like from a very young age, I was always interested in science, both science and science fiction. Actually, like these are two passions of mine that kind of grew in synergy as I got older.
Very, very fun. I can
Mhm.
totally relate to that actually. So right now you are a PhD student in molecular biology. For people who are not scientists, how would you describe what molecular biology is?
So molecular biology is the study of all the mechanisms that happen within our cells. So it goes from it includes genetics, anything that our cell do in terms of development, for example, or also what we call metabolomics. So the study of the how our cells deal with energy and yeah, it's very general, but anything microscopic that happens in our cells. Um, is falls within the realm of molecular biology, basically.
And you study strictly humans?
Mostly humans, yeah.
Mostly.
I or yeah, I so my PhD is very centered around gene editing. So we use tools that are from different species, mostly bacteria, and I guess we'll get to that later with CRISPR. But we try to apply that mostly to human biology. Yes.
Okay. That's very, very intriguing. How did you decide to study that?
So I guess most people heard about CRISPR by now, since there was a Nobel Prize last year for its two discoverers, Emmanuel Charpentier and Jennifer Doudna. And so they characterized the system in 2012, and that was also the first year of my undergrad studies in biology, actually. So I was very lucky to discover this field at its very beginning, and to also to grow as a scientist and to see this completely new field basically explode within the past 10 years almost. Um, so yeah, my first experiment in the lab was with CRISPR, and I kind of stuck to it.
Okay, so before we get to CRISPR, maybe we should talk a little bit about the human genome,
Yeah.
because it's my understanding that in order to use CRISPR, you need to understand the human genome, right?
Yes.
Okay, so and it's my understanding that we've completely mapped out the human genome, or do we still have a lot of work left to do on that?
Yeah, so it's a bit complicated because the first human genome, the first draft, was published in 2001, and it's pretty accurate and it's pretty complete already. But there have been multiple updates since, just because some areas of the genome are a bit complex, they're a bit repetitive, so they are a bit difficult to reconstruct. But actually, the first version of the human genome that's considered as complete, that's like the first end-to-end version, to be simple, was published only last year. So yeah, we're still updating it from time to time.
Okay, and again, for the lay people out there, the human genome is my understanding is all the genes that make up a human being. Is that a pretty accurate statement?
Yes, it's all the genes, and it's also all their regulatory sequences and anything that goes around these genes, which is very complex. But to make it simple, I'd say the genome is just all the genetic information that's encoded in the DNA that's in our cells. So if you take the whole DNA in one cell of our body, you have basically your whole genome in there.
There's something about this, and I forget the actual term, and maybe you will know by just me describing it here. But whenever I think of genes, I think about the research being done um, on how genes perhaps might have memories from
Oh,
granddaughter to mother to daughter. What is that called? Do you know what I'm talking about?
yeah, yeah. So that's epigenetics.
Yes.
So it's a completely different field. It's also very new, and yeah, it's very exciting. So it kind of so basically, if we consider the genome as just the sequence of DNA, the four letters that compose DNA that form the whole genome. Epigenetics are all the kind of chemical modifications on DNA that change the expression of our gene. So it's an additional layer of information that's not encoded in the letters themselves, but in the way these letters are used or are expressed in different cells of our body.
Okay, and that's a completely different field from what I gather,
Yeah.
from what you study, but it is pretty exciting, isn't it? I mean, do you have any thoughts to share on epigenetics?
It is. It's amazing. It kind of it has almost so as you said, epigenetics is a bit different from classical genetics because it can change during our lifetime, where we consider that we inherit half of our DNA from each of our parents, and this is something that stays pretty much constant throughout our lifetime. But with epigenetics, these modifications and different regulations, they can evolve during our lives, and we can still transmit them to our children.
Yeah, it's so interesting that's kind of the things that I find that I have been reading about myself because for me genetics I find is such a I remember studying it in I think it was the science of psychology maybe I don't
Mm.
remember but I remember studying genetics and I hated it. I'll be honest with you, Olivier. It was so mathematical. It felt mathematical. It felt very dry as a subject. It's not like studying tardigrades, for example. But it is, once I guess people who do understand it, you must see the human body in a way that the average person doesn't.
Yeah, I can relate to what you say because, like, classical genetics, which is just studying the way genes are passed on, is very mathematical. And of course, I'm a biologist, so I'm terrible in mathematics. So clearly, I can relate to what you say. Um,
Yeah.
but yeah, I think what I find very exciting about it is that it's very new. Like, in the end, the structure of DNA, the very well known double helix that everyone knows about, has only been discovered in the 50s, I think, or the 60s, right? So it's a very young field compared to, I don't know, chemistry or physics, or these are not things that have been studied for centuries. So it's still kind of it feels really like confronting yourself to this giant of nature with amazing complexity, and it's very humbling, but it's also fascinating in many ways.
So in trying to piece your research together here, you keep saying classical genetics. So is that because that's what you focus on?
No, so I was saying classical genetics as a kind of in opposition to what we do now, which is more basically for many years, genetics was just looking at families, for example, with genetic diseases, and looking at how genetic traits were passed on from parents to children, or in a family tree, how different genes or characters were passed on to certain people, expressed in certain people and not others. I think now with CRISPR and other advances in genome editing, the our approach is a bit different. Now we can go directly start from the genes, especially now that we have the sequence from the human genome. We can start from the genes, modify these genes directly inside cells, and see what happens. So I think, yeah, there's kind of a paradigm shift in the way we approach genetics these days.
Right. So it's less of a, I suppose, less of a not observation and more like we
Yeah,
can actually manipulate these things now.
yeah, exactly.
Wow. And just so that we really kind of, simplify genes, I'll give you an example. I did my 23andMe.
Oh
Right?
yeah.
So the DNA test that where, you kind of looks a little bit at your ancestry, which, by the
Yeah.
way, I'm mostly all French, no
Okay.
surprise. But there is a section in there, and I don't know how accurate it is, but there is a section in there that even says things like, "At what time do you usually wake up? Well, you're probably a late riser," and that it was, like, so accurate, like,
Oh, okay. Interesting.
can you wiggle your ears? Can you it has all these, like, I guess these things are genetic, right?
Well, part of it are, of course, I think it's not something. It's a very big question in biology. And even I'd say in philosophy, what's essential, like what's part of what we are, and what's just acquired through our life experiences, right? And I think now the consensus is that. Our genes of course define who we are, but they also interact all the time with our environment, right? And so this goes back to what we were saying about epigenetics, is that for example, you can have a genetic background that would maybe push you to, I don't know, maybe take more risk as a child, right? Or be, I don't know, more adventurous, let's say, to to simplify, right? But then the experiences in life that you will acquire by doing this will also kind of give a feedback to the expression of your genes. And for example, if you, I don't know, like if you fall from your bike as a young kid, it might completely change your approach to taking risk in the future. So there's this constant back and forth feedback loop interactions between your genes and the environment. And so from about 23andMe, the important thing to know about that is that they don't really sequence DNA. Like they don't really sequence gene and look at their function. They look at specific markers. So we know that some very small regions in our DNA, they are associated with certain traits, but there's no causal relationship between these. It's basically just statistical association of, like, if you have this specific marker, then you are probably from France or from Europe, and maybe these are associated also with statistically with some personality traits. But it's clearly not an exact science.
Right, of course. I'm curious, Olivier, what are some genes that are absolutely hard coded, environment cannot change them, like maybe diseases, things like that?
Yeah, so I think in general, because of the laws of evolution, genes that are very essential to our cells, that are like indispensable for their function, evolve a lot slower, because a tiny change in these genes can completely perturb the whole functioning of the cell, and kill it basically. So I work a bit on cancer associated genes, and one of the most important genes for cancer development is called P53. And we know that in this gene, like some very small mutation can completely change the way our cells proliferate, and this could lead to the development of cancer, for example. So these genes are a bit more, yeah, rigid in their evolution compared to genes, for example, related to, I don't know, the color of the eyes or the color of hairs. These are genes that can evolve a bit more freely because they don't directly impact the survival of their career.
Okay, but like something like cystic fibrosis, for example, which I think is a genetic disease, if I'm not
Yeah.
mistaken,
Mm.
is something that, it doesn't matter what diet you have, it's
Yeah.
a gene that's not gonna change, right?
Well, it can change over time, but of course, because of this interaction with the environment, like I can give the example, for example, of sickle cell disease, because that's something I know a bit more about. So sickle cell disease is a disease of hemoglobin. So it's a mutation in hemoglobin that kind of impacts the structure of the hemoglobin molecule and how and its ability to fixate oxygen in the blood. And so if you so it's very common in people with African ancestry. And the reason for that is because if you only have one copy of these sickle cell gene, or one mutated copy of the gene, because we have our chromosomes always go by pair, so we have two copies of each of our genes. So if you have only one copy of this sickle cell gene, it doesn't cause any adverse effect, and it protects you against malaria, because it prevents malaria is caused by a parasite that gets into your red blood cells. So one of these mutations prevents the parasite from getting into your cells. But if you have two mutated copies of this gene, then it makes you sick, and we know that the symptoms from sickle cell disease are pretty bad. Like, it causes generalized pain and blood clotting. So you can see like how one mutation that could cause a disease in some contexts could give an advantage to people in another context.
That is really, really interesting. So now this begs the question then, could you give people a sickle cell gene in order to combat malaria if they don't have one already, right? Because you don't want to give them two.
Yeah,
Is
yeah.
that something that could be done perhaps in the future?
So that's something that could be done now. And but it's a bit tricky because if that's something that you pass on to your child, then you don't want your child to have the two copies, right? So you kind of have to think about the big picture, and how you don't want the whole population to have one copy of this gene, because otherwise their child will have a greater chance of developing the disease, right? But actually, something that's being done now is the opposite: is trying to, for people who have sickle cell disease, is to take some bone marrow from them, edit it outside of their body to kind of to correct this mutation, um, and then we can just re-inject the corrected cells inside their bone marrow. And we know that just correcting like something like couple percent of the expression of hemoglobin, like 5%, completely changes their quality of life. Like, it goes from it almost they can almost live like a normal life after that.
Okay, wait, we can actually do that today?
Yeah, so there are a couple clinical trials ongoing. I think both in Europe and in the US. So it has been tested on a small number of people, and the results are really amazing. It's really, like, again, you just have these people that are their quality of life is terrible because of this disease, and you just give them a one time treatment, and in theory, it could, like, these cells can just stay there for their whole life, and they are cured basically. So now I
Wow.
think they are trying to scale up these clinical trials to more people.
That makes a lot of sense, and that would be just, life-changing, especially, I would imagine, on the continent of Africa,
Clearly,
where
yeah.
and also it would protect them. Now would they then also have the malaria protection?
Yes, because so yeah, I oversimplified it a bit when I said we corrected the mutation. Actually, what we do is just kind of so there are different types of hemoglobin, right? There are different proteins called hemoglobin that are expressed in our genes, and especially when we are at the fetal stage, uh, we only produce what's called fetal hemoglobin, and then that's something that the gene is kind of turned off when we become adults, and we don't express it anymore, and we express or other types of hemoglobin that carry this sickle cell mutation. And so the goal of the particular clinical trials I've been talking about is to turn back on this fetal hemoglobin gene, so it compensates the lack of the other ones.
Okay, I see. So let's talk about CRISPR now, because CRISPR is the kind of technology that you would use to do this kind of gene editing, correct?
Yes, yeah.
Okay. Uh, what does it look like? Is CRISPR the name of a technique? Is it the name of the machine that does it? What is CRISPR?
So CRISPR, when we think about CRISPR, most of the time we think about CRISPR-Cas9, which was the first CRISPR system that was discovered. But there are many other types. But the most important is CRISPR-Cas9, and so this is a system that's been discovered in bacteria, and it's composed of two components. So one is the Cas9, which is an enzyme, and this enzyme is associated with what we call a guide RNA. So now, thanks to the pandemics and the vaccines, everyone knows what RNA is. In this case, it's not a messenger RNA like in the vaccines. It's a much shorter one, and it's called the guide RNA because when it associates with Cas9, it kind of guides the enzyme to any DNA target that's complementary to the guide. So what happens is that we can design a guide RNA that's complementary to any gene that we are interested in, and the Cas9 will take this guide and scan the DNA, and when it finds the sequence that corresponds to the guide, it will just bind there and cut the DNA.
Okay,
That's pretty much all that CRISPR does.
but is CRISPR is it a physical mechanism?
Yeah, so CRISPR is just the association of these, what we call CRISPR in is the association of these two components, and this is so it's from bacteria, because in bacteria it forms kind of an immune system, a basic immune system in bacteria. So when we say CRISPR, we think about these two components: it's one enzyme and one RNA.
Okay, so in my head, CRISPR was a big machine. Yeah, I had no idea that it was an actual process,
Mm.
more than an actual machine like a telescope or a big microscope. I
It's
really
it's a
honestly thought,
molecular machine. Yeah.
okay, okay, but it's not. It's CRISPR is something that can be taken. It's a process that, from my understanding, now that you've explained it, is a process that you can take from one lab to another as long as you have the equipment.
Exactly. Yes.
Okay, okay, now it makes sense. Most people are scared of CRISPR. I guess most people
Yeah.
don't know that what it is. Now, uh, I guess the question is, why should they not be scared of CRISPR?
Yeah. So I think what's important to understand is that what we do with CRISPR, we've been doing that before with other tools. The big difference that CRISPR made is that it made what we did before a lot easier and a lot faster. Because we use this small RNA component, this small guide RNA is very easy to make and very easy to change. So we were able with other tools in the past to modify DNA and to cut at certain specific sites in our genome, for example. But now with CRISPR, we can just do it much faster and cheaper. And what I would say to people who are afraid of it is that, in my mind, CRISPR is more a tool for research. It had already a huge impact in the past 10 years on genetic research. So it's just like any other tool. Like it's not good or bad. Like technology is never good or bad. It's just, of course, the way you use it, right? Um,
Yeah, I think people they're scared because, people are kind of misled to believe that, for example, genetically modified food is bad for you.
yeah,
And so therefore they associate things like CRISPR and gene editing to like playing God, and that we shouldn't modify natural things, and et cetera, et cetera.
yeah.
And so I guess the argument, I guess, in order to kind of argue with that, one would have to know if there are any side effects to editing genes.
Yes. So there are side effects that we know of with CRISPR. For example, is that for example the scanning mechanism that I was talking about, where Cas9 scans DNA to find the right target. It's not 100% accurate. So sometimes Cas9 can make mistakes and cut at unintended sites in the genome. So that's something that we are very aware of, and that we can learn to we are slowly learning to mitigate. Right. So it's really a work in progress to make these tools always more accurate and safer. But and I also understand why people are skeptical about just the term modifying or genetic modification. Right, it's something that has a very negative connotation, and I can very well understand that. But what's important to know is that, for example, when we think about genetically modified food, we've been genetically modifying crops for centuries. We've been selecting them for specific characteristics. We've been breeding them artificially to get rid of some of their characters and enriching for some others. And something that many people don't know, for example, is that the companies that produce seeds for agriculture, they just routinely produce new crops by irradiating seeds, for example. So they just take seeds from plants they are interested in, and they subject them to radiation, to high doses of radiations, to induce many random mutations in them. And then what they do is they just look at the plants that result from that, and they just select the ones that have the characters that they are interested in. And this is something that's been going on for decades, and that is not considered as genetic modification or as GMOs. But if you just use CRISPR, for example, to introduce the exact same mutation you use with this process, but just to introduce precisely instead of bombarding your seeds with radiations, then it's considered as genetic modification. So I think what's important to understand is that genetic modification is just a tool, and I think in general we should focus more on what we do with it, and what are the applications, what are the characteristics of the plants, for example, that we produce with these, instead of just focusing on the technique we use.
Yeah, that's a very, very, very good point. I'm glad you brought up that example. Can CRISPR be used for cloning?
So it's interesting because cloning in molecular biology has a kind of a different sense as compared to what people think of when we say cloning. So I guess by cloning you mean just making two
Copies.
identical
Yeah.
people, right? So actually, to do that, you don't even need CRISPR, right? Like the clonings of animals have been performed way before CRISPR was discovered. So to do cloning, what you do is just you take DNA from an adult human or animal, and you get that take their whole genome, their whole DNA, and you put it back inside an egg cell, and then you hope that this will produce an identical individual.
But on the molecular level, could you use CRISPR to, like, copy a gene? Is that something
Uh,
that could be done?
yes. So what I was like, as I explained before, what CRISPR does is just cut DNA, and that's the end of the process for CRISPR. But our cells really hate DNA breaks because these are toxic. They are dangerous for the integrity of the cell. So when there's a DNA break, and this happens routinely, like we have DNA break in our cells every day when we get in the sun. It's a pretty normal process, right? And this is why our cells, they have many mechanisms in place to repair these breaks as fast as possible. And so when you have a DNA break, our cells can just stitch back the two pieces together and nothing happens. But many times this process it doesn't work perfectly. So it just introduces random mutations at the cut site, and when that happens in the middle of the gene, many times just it just completely disrupts the function of the gene. It destroys it completely. So with CRISPR, we can delete a gene by doing that, but we can also insert a new gene, as you were saying, by still making the same cut inside in the DNA, but by also providing the cells with a repair template. So if we provide a piece of DNA to our cells that looks like the damage site, but that contains a mutation of interest, for example, or a new gene, uh, our cells will just take this template and use it as a model for the repair. So in that way, we can introduce, for example, a completely new gene or a precise mutation in our cells.
So cool. I'm listening to you talk about this, and I have like big wide eyes because I'm like, oh, okay, now I'm starting to love genes. You know what I mean? And so now, tell me a little bit about your own research. You mentioned that you're working a little bit with sickle cell. I saw on one of your biographies on a website that you've worked with virus production, gene editing, genetic mutations. Tell me about the research that you're doing.
Yeah. So the general topic of my PhD is more in the direction of technology development. So what I'm trying to do is to develop new tools based around CRISPR, that are more efficient or more precise uh, to mess with our DNA, basically, and then use these tools to study disease mechanisms. So, for example, I was saying that you can just destroy a gene by just cutting with CRISPR in the middle of this gene and introducing random mutations that will disrupt the function of the gene, right? And by doing that, we can study we can study different genes that either we know they are either we identified them and we don't know what they do, or we know what they do in certain cell types but not in others. And so by simply cutting with Cas9 in the middle of a gene in cells in culture, for example, we can just destroy a specific gene and then look at the cells and how they behave. And that gives us a pretty good hint about the function of this gene. So it's a very naive approach of just breaking stuff and see what happens, basically. And in the context of diseases, it's something that's very useful. For example, in cancer, you can just destroy many genes with that method in cells in culture and see which ones become cancerous. And that gives you a pretty good indication that the genes you messed up with are important to prevent the apparition of cancer. And one last thing that we are working on, that we've been working on recently, is with SARS-CoV-2, the virus at the origin of the COVID-19 pandemics. We can also just take cells from the lungs that are normally infected by the virus. And by destroying different genes in the cell, we can see at what point these cells cannot be infected by the virus anymore. And so when that happens, when we find a gene that when it's broken, the cells become resistant to the virus, it also gives us a pretty good indication that this gene is hijacked by the virus to infect us. And so this could potentially lead to finding new treatments against the infection.
Okay, so first of all, I didn't know that viruses could infect genes.
No, so viruses infect cells, right? But to do
Okay.
that, they need to penetrate cells. They yeah they need to get into cells and inject their genetic materials inside the cell, and after that they just hijack the whole cell machinery to replicate themselves. They are parasites in some way. But to get inside the cells, the virus, like we heard a bit about the spike protein of the virus. If you think about the SARS-CoV-2 virus, the images we see is that it's kind of a sphere, and you have these spikes around it. And these are proteins that they act kind of like a key, and when they are complementary to some receptors that are the surface of our cells. So our cells, they just communicate with each other, they just communicate with their surroundings all the time, and they have receptors at their surface to get information from outside. And what the virus does is that it has this spike at its surface that binds some receptors, and they act like a key, basically. So they just bind to a specific receptor at the surface of the cell, and it gives the signal to the cell to let them in. And so what we are looking for by looking for we are looking for the genes that express these specific receptors at the surface of cells.
Wow. So have you actually looked at the SARS-CoV-2?
Yeah, we're working with it right now.
Oh, cool. That's really, really neat. So I was really curious because a lot of the stuff that you're doing is medical in nature. It is,
Yeah.
it really could impact our future. Where do you see this technology going? I mean, like you said, you're researching technology right now. You're trying to find better solutions. What do you hope for in the next 5 years?
Yeah. So as I said, I think CRISPR already had a huge impact on in research, and there are still new tools that that are getting developed almost every month. It's really a crazy field. It's evolving insanely fast. So 5 years actually in this field is it feels like centuries. Really, it's impossible to predict what's going to happen in 5 years. But some things I'm pretty excited about are new technologies that instead of just simply cutting DNA and letting the cell do the job to repair, there are new systems that are being developed to instead create more precise mutations and clearly. There are teams in the US, for example, that that developed systems that can just modify the letters in the genetic code instead of just breaking it. So in their case, you don't cut DNA. You just have an enzyme that's going to change an A with a T, for example. And so it's much more precise, and it's also much safer because you don't introduce these kind of toxic DNA breaks. And I think, yeah, I'm very optimistic that we're gonna get to systems that are very precise, very safe, that we can tightly control, and that can introduce any type of mutation anywhere we want, and then we can just safely use these in patients to correct potential mutations.
The Atlantic published an article, I think in December last year, or maybe the year before, about the possibility of eliminating Down syndrome,
Yeah.
which is a very large ethical question that we have to deal with. Is it ethical? Is it something we should do? Is it, it's a very, it just really is a huge question. As a research scientist, do you have boundaries? Are there things in terms of ethics that you would never do, or are there things that you're excited in doing that are perhaps controversial? Like, where would you draw the line?
Yeah, it's a huge question. It's really a fascinating question. But I think that's something. This is really a conversation we should have as a society, and I don't think like I, as a scientist, should be the one to make this decision for people. As you said, Down syndrome is one example, but you have a even grayer area as well. Like, for example, people with who are deaf for genetic reasons. Many of them don't even consider it as a disease, actually, or as a handicap, because they have their own, their whole family environment is they are surrounded by people who also have impaired hearing, and so some of them actually, or probably if you offer them a treatment, some of them probably wouldn't even accept it, right? So I think um, I'd say in the field right now, the biggest taboo is really embryo editing, which is instead of correcting a mutation in an adult who can give a consent and who can agree to receiving a treatment, you can also perform this modification at the embryonic level. Um, and yeah, it's really a gray area because you are dealing with the DNA of someone who's not even born, who cannot give consent, and the mutations you introduce, they will also be passed on to this person's childs. So then, yeah, it really becomes it feels kind of opening the Pandora box when you start modifying DNA in embryos. That's something I wouldn't do personally.
Yeah, I wrote a paper in high school. So we're talking like 1990s
Mhm.
about eugenics because that was a big thing in especially in Britain. And I can't remember why, but I think that they were really, really having the debate about eugenics. Um, and I think this really touches on that topic. You indicated, it's funny because we actually met through a science communication conference,
Mhm.
and you indicate on your Twitter account that you're big in science communication. So, what is it about that you're passionate about?
Yeah, so actually, I'm not even remotely doing as much science communication as I'd love to do. Of course, a PhD is very time consuming. It's very stressful many times. So I would love to do more, but I'm also very glad that working in research gives me the opportunity to do this. So I think it's pretty obvious now with the pandemics why we need more and better science communication. I think what's really striking to me is that people are actually eager to learn more about science. And even if you look at like conspiracy theorists around vaccines, around COVID, around yeah, I don't know, flat Earth, these are people that are actually getting by themselves pretty deep into the science, and they don't do it the right way, they don't reach the right conclusions for sure, but they actually have the motivation to learn a lot about very complex and specific topics. Like I was very stricken when I watched this Netflix documentary about the flat earth society. I think it's called Beyond the Curve. And so you have these people that are basically trying to scientifically demonstrate that the earth is flat. So they do actual math and they design legit experiments to try to demonstrate that the Earth is flat. And unsurprisingly, they keep redemonstrating that the Earth is not flat. But I think it's kind of impressive for just lay people to be able to get into such complex topics because, I mean, I know the Earth is not flat, but I don't know why. I wouldn't be able to demonstrate it by myself, right? Um, so to go back to science communication, I think there was this conception in science communication for a long time, which was called the knowledge deficit model, where people thought that basically people didn't know enough about science, and the more information you would give them, the more they would trust science. And we know now that that's not true. I think as scientists and researchers in particular, we like to see ourselves as carriers of objective truth. But I think, especially in the context of a pandemic where many information are circulating, we should not like see ourselves as independent actors that are separate from the public debate. I think actually that scientists should um, get politicized in some way, or at least like take a stand in the public debate, like understand that science is political, anything we do is political, especially in fields like CRISPR, for example, that are controversial and that raise very deep ethical questions. And I think we should accept that, and we should get a bit more involved, and yeah, just explain to the public what we do and how we do it.
You bring up a really good point in that, one of the last interviews I did where I was a guest, I was asked if if I felt like there wasn't enough science literacy in the
Mm.
public, and that's why we have conspiracy theorists, and I very much disagreed, and I really feel as a non-scientist, but who's a science communicator, that the problem is the lack of trust.
Yeah.
There's a lack of trust in the institutions, including medicine, science, the academy, and all that stuff. And I think that there's, like you said, this kind of sometimes ivory tower looking
Yeah.
down on people. Do you feel that sometimes there is a disconnect between academia and non-academics?
Oh, definitely. And I think it's a complex question because, as you said, there's a lack of trust in scientific institutions, but it's not limited to science, right? I think in the Western world now, there's a big lack of trust in um, what one would call the establishment, and we have to accept that we are part of it. Like, and universities are very big and very bureaucratic institutions. And so, yeah, I think it's a general issue that scientists alone cannot solve the trust issues in the institution, of course. um but I think, yeah, as a researcher, I feel clearly there's a lack of support, for example, first of all, for these kind of initiatives. It's starting, I have to say, in many universities now, there are, like, university cities are starting to recognize that communicating about your results is actually part of your job as a researcher, because you work with taxpayer money. So it's only fair that that the people you work for get the information or about what you discovered and what you are working on. And I think there's also sometimes sort of a feeling of helplessness from scientists, and that's at least how I felt at the beginning of the pandemics, where you see all this false information circulating, and it feels like just too much to fight against. And yeah, clearly there's a lot of work to do, and I think we spent too long just being a bit passive about that. And I think universities should become again the place of open knowledge, and of course open access, for example, for scientific publications would be one very important step in that direction.
That's a really, really big one is the journals. Uh, again, as a non-scientist who does science communication about microscopic life,
Hmm.
I don't know everything, and sometimes
Yeah.
I want to access a journal article, and because I'm not associated to a university, I don't have access to them. So it can be extremely difficult. We have about 10 minutes left. Olivier, I really
Hmm.
want to talk about your other hobbies outside of
Hmm.
science. I see that you play the bass guitar.
Yeah.
Tell me
Yeah.
about that.
So yeah, I used to sing in a band for a long time, but I have to say I'm not really a musician by training, so it's more of a just a naive hobby. But yeah, I've always been a fan of music, of heavy music in general, like metal, rock, blues. I've always loved that, and so I got a chance when I was younger to get into a band where they really needed a singer, and so their standards were low enough for me to get in. And more recently, I got into bass. Yeah, I play in a new band uh, called Monoatomic God, and yeah, I play bass there.
Are you guys all scientists?
No, 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 I'm the only one.
Okay. I had to ask because of the band name.
Yeah, yeah. I also write the lyrics, so I kind of inject a bit of science in there. I feel
Oh, so you
like
guys create you guys create your own original music.
yeah, 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 yeah. We
Sweet.
write only original songs. But yeah, I feel like art and science mix very well in general, and it's probably because art and, as I was saying at the very beginning, like science fiction was probably the thing that got me into science. I really like closing the loop now and re-injecting some of the science I do inside music.
Absolutely. Now, science fiction, do you prefer books or films?
I think in science fiction, I read more books. I grew up with the X Files, and it was it was very nice recently when like there was this last season that came out like 10 years after the end of the series, and like in the last season actually they justify, spoiler alert, but they justify everything that happened in the series with CRISPR. Actually, they like the final
Ah,
plot twist is that the government used CRISPR to inject alien DNA in our genome. That was pretty cool as a scientist to see that.
wow. That is actually really cool. Hey, actually, do you think that could happen in the future?
Well, we need to find alien DNA first,
Well, I mean, we
but
are going to. I think it's
hopefully.
funny. It's funny because I've interviewed a couple of, astrophysicists
Mm,
slash astro biologists, and it's something that I always ask them. I'm like, you guys study space. Do you think there's alien life? And they all say yes. I mean,
yeah,
it's inevitable. There's so much out there that
yeah.
mm,
Actually, it's one of my scientific dream would be to get my hands on some alien DNA or whatever it is. Actually, because the interesting question about that is that if we find extraterrestrial life, like we could imagine that they would have a completely different form of support of information that could be completely different from DNA. Right? I saw papers like where people talk about carbon based polymers or these kind of things. And actually, I don't know what would be the most exciting. Like if we find something that's completely different, or if we find a form of life that also has DNA, and in the end, that is kind of similar to us, because that would just mean that maybe this is the only way to. This is the only way for life to exist. Right? I think in both cases it would be amazing.
I think that's the big question that's going to be answered, probably in our lifetime, if I were to guess.
Hopefully.
Especially because they're bringing back soil from Mars.
Hmm.
I'm betting on the fact that we're gonna find, like, a Martian tardigrade.
Oh yeah, but and these guys are alien enough, right? Like
Oh my god, yes.
that's the thing. Like we have, I think that's what I like about biology is that it's a pretty young science. It feels like there's still so much to discover, and sometimes some organism on Earth feels completely alien, really. And so yeah, we still have a lot of work. We still have a lot of stuff to pass the time before we find something on Mars, I think.
Yeah, and I mean, just the creatures in the deep sea, right? I mean, we still
Hmm.
don't know a lot about what lives really, really, really
Oh
at the
yeah.
bottom of the ocean.
Yeah, yeah, clearly. Also, like just yeah, sometime when I have time, I like to read research articles about octopi, for example, and they are like that's just crazy, really. When you study the human genome, it becomes as you said, you get a different perspective on our body, on how we perceive what happens in our body, right? But then so it becomes familiar, but then when you look at these yeah weird organisms, it's like all paradigms are just shattered. It's completely different.
Yeah, it's really cool. So I guess to close up the this episode, I wanted to know what are you doing after you're done your PhD? Are you staying in Switzerland? Do you want to go to the United States? What's the next plan?
So actually, before starting my PhD, I was working for 1 year in a company in California. So I'd really like to go back to the industry for some years because I feel like the biotech industry, the way it works, it I feel is a lot more collaborative. Like I really enjoyed working in companies because I was interacting all day with informaticians, data scientists, chemists, and I feel like it was a very exciting environment for me. Um, but then also, as I said, I would also really like to do more science communication. So when I have a bit more time, I'd really like to start maybe a Twitch channel. I think that's something that I'm really attracted to because I tend to listen to a lot of podcasts just like the one you do, or yeah, long format interviews. So yeah, that's something I'd like to explore.
That would be really cool. There's a lot of us on Twitch, so come and join us and come to the dark side. It's a lot of fun. Olivier Belli, again, thank you so much for being on the show. I really learned a lot from you. I actually can't believe that an hour has already gone by. I could
Yeah.
probably ask you 200 more questions, but we'll leave it at that for now. Good luck with the rest of your studies, and I hope that do you. So, is your band somewhere on Instagram, or is there anywhere I can listen to your music?
Oh, so we are on Bandcamp. So, but
Bandcamp.
yeah, you can find the name on my Twitter. Actually, it's Monoatomic God.
Cool,
Yeah.
cool. So we'll link to your Twitter account then.
Yeah.
And again, thanks for coming on the program.
Yeah, thank you very much. It was a lot of fun, and good luck for your future podcast. I'm looking forward to listening to.
Thanks very much.